Good afternoon, everyone. Is my audio clear? Can somebody message?
Yes, sir, it is clear.
Okay, thanks. So, in the last class, we were discussing about DNA polymerases of E. coli, and I mentioned that there are different types of DNA polymerases, like DNA polymerase 1, 2, 3, 4, and 5. So, in that, I did mention several things. I wanted you to remember what is meant by what is the rate of polymerization, how fast can it do, and different DNA polymerases have different speeds. Okay, and there is also one more thing that we wanted to know is their ability for proofreading, that is accuracy. Right? So any actions or work will depend on speed and accuracy. Right? And there are also two more DNA polymerases called 6 and 5. Sorry, called 4 and 5. And they are mostly involved in DNA repair. It's called as translation DNA synthesis. We will discuss about it in the future classes when we get to proofreading and so on, or mutations and repair and so on. What I want you to here understand is the speed. I hope you remember, it's a general intuitively, speed is inversely proportional to accuracy, right? When we try to do something very fast, we are likely to make more mistakes. And efficiency then becomes that how much of correct work are we able to do in a given time. So we are all in a struggle for efficiency, right? Which includes these two things, speed and accuracy. Speed is this polymerization rate. How many bases can it add per second? So if you look at DNA polymerase 3, you notice that it can add up to 1,000 bases per second, whereas polymerase 1 and 2, they can do so much. And if you look at proofreading activity, we will count both these things. Accuracy is the ability to add correctly, that is fine. And if there is a mistake, are you able to correct it or not? What we see is DNA polymerase 1 can correct more mistakes, whereas DNA polymerase cannot do as much. DNA polymerase 2, they all have different specialized functions. If you look at, and also I would want you to appreciate what is processivity, that is number of bases it can add or subunits it can add before dissociating from the something like primer template junction. DNA polymerase 3 has a huge processivity compared to that of polymerase 2 and 3. If you look at DNA polymerase 4 and 5, they don't have speed, they don't have accuracy, and they don't have processivity. Yes, I logged out and logged in again, so I couldn't. I hope you can see that now, right? Thanks for the message about the thing. Yes, so I was talking about DNA polymerase 4 and 5. DNA polymerase 4 and 5, they do not have speed, they do not have accuracy, they do not have processivity. And still, they are useful to the cell, right? And that is what they are useful in something called as translation DNA synthesis, which we will discuss later. So some of the proteins that are involved with in replication or what we have discussed, I discussed about processivity, and we also discussed about exonuclease activity. There are two types of exonucleases activity: 5' to 3' exonuclease activity, and 3' to 5' exonuclease activity. So if there is a DNA like this, assume this is 5' and this is 3', this would be 3' and this is 5'. And if there was a wrong base added here, and it should be removed, and then the removal of it is being done by, I hope you can read that it is done by 3' to 5' exonuclease activity. DNA polymerase moves back a little bit and makes the correction there. It will just delete the part like that. That is 3' to 5' exonuclease activity. There are also DNA polymerases like DNA polymerase 1 has 5' to 3' exonuclease activity. Assume that there is a DNA here, which would be 3' to 5'. And DNA polymerase was synthesizing, and it has to remove these parts here. Then the polymerase exonuclease activity used there is 5' to 3' exonuclease activity. What it would do is, one basis at a time, it will keep on cutting something like this, right? It is using 5' to 3' exonuclease activity. Of the three DNA polymerases that we discussed a little while ago, only DNA polymerase 1 has both. DNA polymerase 1 is kind of special because it has 5' to 3' polymerization activity. Okay, it has 3' to 5' exonuclease activity like most of the polymerases, and it also has an additional 5' to 3' exonuclease activity. So it can do many more things, and it is also involved in repair and other activities of the cell. I hope you also remember from here, this is the site where the active site for polymerization is. And here is the active site for exonuclease activity. If there is a misincorporation, there is a mismatch, because of which the DNA might slide to the exonuclease activity, and there some of the bases would be removed on. And then once it is removed, mismatches are no longer there in the DNA, so it will go back to the active site for polymerization, and then polymerization would occur. So this is the same thing, repetitive. So I will move. Sorry if you are hearing the ding sound. For some reason, I don't know why. I had to admit everyone into the classroom, not by automatic. If something is wrong, I'll try to check it, rectify it later. And again, in the last class, we also discussed about direction of replication, about polymerases, about Okazaki fragments, and so on. In today's class, the most important part is the ability, or we shall try to draw the diagrams for bidirectional replication. Okay. Typically, in all CA exams or even in the final exam, I usually ask people to draw the diagram of a bidirectional or replication fork. So we'll use this base and assume that there is a DNA molecule, okay, circular DNA molecule. I'm going to erase this again, but a DNA polymerase replication has to start at one point. That means the DNA at one place should open up something like this, right? It should open up, and then new bases would be added like this in this direction. So this is one replication fork going this way, and this is another replication. You have two different DNA replication forks. All would have started at one point that is usually called as origin of replication. Okay, so I'll erase this, and let's see if we can draw how things would be, where the objective is to represent all the DNA strands. And always, as I told you before, we should we should label everything properly. So if you are interested, I would suggest that you draw along with me, because that will give you a good practice about when you want to reproduce similar diagrams in exams. Okay, I have DNA molecule, and I'm going to draw a small line to differentiate that this is to differentiate the two replication fork. The yellow dotted line is where exactly the replication fork would have started replication. The region here, okay, I'll also have to put 5' to 3' always, 5'. So the other end would be 3', 3' OH, and 5' phosphate. The complementary strand would be anti-parallel, therefore it should be 5' to 3'. Okay, and the yellow line is representing the origin of replication, simply denoted as ORI. Now I have to draw two replication forks. This is one that I'm showing here, and the other one is here. So replication always happens from, polymerization always happens from 5' to 3' direction. So if I put a primer, here, like this, then replication would have happened in this direction. This is, this would be the 5', and this is the 3'. And polymerization would have synthesized this DNA like this, would keep on synthesizing. And because it is continuously produced, we would call it as leading strand. It's produced continuously. What about the bottom strand? Polymerization would have happened in the reverse direction. So what we would have had is something like this. This would be the primer. Primer is made by the enzyme primase. This would be 5', and this is 3'. And the polymerization would have occurred something like this. Similarly, this strand is synthesized discontinuously, and therefore it is called as lagging strand. And each of these fragments are called as each of them are called as Okazaki fragments. The RNA primer is produced by primase. Okay, and I also want you to, just for understanding sake, there is a here, if you see, this part of the blue line is indicating DNA, and the red one is indicating RNA and their base pair. That is the hybrid region. I'm just indicating it so that when you know at the end the RNA primers are removed by RNase H. H stands for hybrid. So it removes RNA wherever there is a hybrid of DNA and RNA strands. Now let's look at the other strand. What would be there? Now this strand would be, the primer would be like this, and because it would be 5', and this is 3'. Right? And it would be, polymerization would have occurred continuously like this. And here, in this case, this is the leading strand. And if you look at for the opposite one, it would have been the something like this primer, and there will be another primer here, and polymerization would have occurred in this direction like this. Okay, So this summarizes, well, these are here you would have been having this hydrogen bonds. Okay, this is the double stranded region, and here you had denaturation. This whole structure here it is showing denaturation, right? That is the origin of replication. And then new primer is synthesized like this. These two will be the first synthesized primers, and DNA polymerase will start synthesizing. And so there is one replication fork arranged here on this side, and one replication fork arranged on the other side. So there are two DNA polymerases at any given time. One DNA polymerase synthesizing the leading strand here, and one polymerase synthesizing the lagging strand on this replication fork. So I will just number them so that it would be easy. I will call it as replication fork 1 and replication fork 2. And on the others, at replication fork 2, you have leading strand this side, whereas on replication fork, this is the leading strand. It all matters about, if you remember 5' to 3', anti-parallel, and how you should do it, then everything will be, you can then draw this replication for any given time. Okay. So RNase H, I already described just now that it's the RNase which will remove the RNA. And DNA polymerase, like DNA polymerase 1, will fill up the gap, and the nick is something that is filled up by DNA ligase. Helicase is the enzyme that denatures the DNA, which should open the replicate at the origin of replication. It will start, and then it will keep on removing or breaking the hydrogen bonds between the two strands, thereby allowing it to become single-stranded. Only then DNA polymerase can polymerize on that level. So one more thing is, it's usually a hexameric one, and it moves from 5' to 3' direction. Everywhere you should always keep this 5' to 3' direction. You can think of polymerization or transcription, or the ribosome reading the mRNA, or even the helicase moving on the on DNA. It's always in the 5' to 3' direction. Okay, and you should also remember that it utilizes energy. ATP is required for breaking the hydrogen bonds. And then single strand binding proteins. So it's something like helicase has worked, has consumed energy, broken the hydrogen bonds, denatured the DNA, and then if you leave it like that, DNA will reform or renature on the hydrogen bonds again. So there are single strand binding proteins which will coat the single strands so that they do not form, they do not go on renature. Then topoisomerases, as we say, I'll try to draw. I hope you will get it, but otherwise we could have done it as a model if we were in the class. But we'll just try it here. Okay, count how many twists are there. I'll just draw it like this. What is the twist? What is twist? Every time one strand crosses over another one. So this is, I'm looking at, I will just go by different colors for clarity. 1, 2, 3, something like that. This is one strand. It will go, and 2 and 3. Count just as 3 is the twist, just for understanding. Now, if one were holding this strand and another was holding, and they pull them apart like this, okay, pretty differently. If this strand is pulled like this and this strand is pulled like this, something trying to open up or denature the DNA, what would happen then? I assume that this side it is fixed, okay? Then it would become something like this. It is open. It will become like this. One, two, three, something like that. Okay. Here, if you look at this, the DNA amount of DNA, number of base pairs is same, right? So assume that it has 30 bases. So its twist will be 3, right? Number of times one strand will cross over the other. If you are pulling these two strands apart, the number of bases is 30, but you have high tension. You have made it. You have 3 twists within this much amount of DNA. Okay. If you keep on pulling, what would happen? Somewhere some strand is going to break, and then it's going to degrade the DNA, right? To ease this tension, DNA nature has come up with enzymes that we have already discussed about DNA topoisomerases. Topoisomerase 1 and topoisomerase 2. Topoisomerase 2 makes double strand breaks, and then it relaxes the supercoiling tension or potential energy. So here we have, because of that pulling like action, you have positive supercoils that have formed, right? And that is eased by topoisomerase 2. It makes a double strand break and it will reseal. So every time it does that, it reduces the supercoiling by in steps of 2. Okay, the positive supercoil is now brought into negative supercoiling. That's the role of the topoisomerases. You might find this everywhere with regard to transcription and most importantly with replication. So we have to also mind that there are differences between prokaryotes and eukaryotes in terms of the DNA polymerases. Prokaryotes or something like E. coli has about 5 or so DNA polymerases. That is called 1, 2, and 3 that we have been discussing. You'll count the same as the holoenzyme. And then we have called 4 and called 5. These two are involved in replication, in errors, repair mechanisms. Call 2 is also in repair. Call 1, DNA polymerase 1, can also remove the RNA primer. Hope you remember what I told you about this. Say, assume this is the RNA primer, okay, of one Okazaki fragment, and this is the DNA that is being synthesized. DNA polymerase 3, which is usually the one that is involved in chromosome replication, will synthesize. Say, assume there is a primer here, it's synthesized till here. Here you have another DNA, RNA molecule base pair to DNA like this. So it will stop and it will release the DNA. Then that place, instead of RNase H can also remove the primers, RNA primers. But DNA polymerase 1 also has the capacity to remove the RNA primer. And what exonuclease activity is it using? It will use 5' to 3' exonuclease activity. So bit by bit, it will keep on deleting the RNA like this, and it will keep on synthesizing the new DNA like this. And eventually, this gap is sealed by lipase. Okay. In eukaryotes, you will see that there's a huge number of DNA replication, DNA polymerases. So here you call it as DNA polymerase alpha, which also alpha DNA polymerase has something like this, and it also is attached with a small RNA primase-like thing, primase-like enzyme. So it is used in synthesis of primer and then initiating the DNA synthesis. Then there are many different DNA polymerases that are used in replication and also in repair and so on. These two are something that you need to keep in mind, called delta epsilon. Okay, delta will synthesize lagging strand DNA synthesis, and also is involved in repair mechanisms. The leading strand is done by epsilon. Okay, so in replication, these delta and epsilon are involved. So you need alpha polymerase called alpha to initiate the replication primers and so on. Then we have delta for lagging strand, and we have epsilon for leading strand. Okay? These three are important for replication in eukaryotes. There is polymerase switching, you call that. So how does initiation happen in eukaryotes? So you have this DNA polymerase alpha primase complex. It comes as a, see, this is the polymerase and this is the primase part of it. So it binds and synthesizes a little bit of primer. The green one shown is the primer, RNA primer. That is taken up by the alpha and synthesizes this orange part is the primer, DNA primer. And then either DNA polymerase delta or epsilon would come along with the sliding clamp. We haven't discussed about sliding clamp. Sliding clamp is responsible for processivity. What is processivity? Number of bases added before dissociation, right? So because what does the sliding clamp do? It encircles the double strand DNA like this, as shown here. And then because of which sliding clamp is encircling the double strand DNA and DNA polymerase is attached to the sliding clamp. So it will remain, it is like sliding clamp tethers the DNA polymerase to the substrate or primer template junction here. And therefore it will not dissociate and keep on adding a lot of DNA, a lot of bases to the growing strand. So in sliding clamp in eukaryotes is also called as pCNA, proliferating cell nuclear antigen. I think this particular protein was produced or found often in cells that are proliferating. If cells are proliferating, what should precede it? Replication. In cells that are actively proliferating, replication will be seen. Because replication is there is some new protein that is being observed in that particular, in the time of discovery, okay? So that they named as pCNA, proliferating cell nuclear antigen. This is the sliding clamp. Yes, we will get to the mechanisms of replication now. And I want you to, I hope you have already watched the video and videos that are there about from Watson videos. Please watch them multiple times. So there is a holoenzyme. Holoenzyme is kind of the complex multi-subunit, multi-enzyme complex, which has many things in it. Here, if you see, this is the helicase, which is responsible for separating the DNA strands or denaturing the DNA strands. Here you're seeing the, this is the clamp, sliding clamp. Here is another sliding clamp. We will get to, in the last class, I have shown you a nice, beautiful video. I hope you have watched it. You could always search for. There are excellent videos on that. There is tau protein, there is gamma, there is delta. And these are the DNA polymerases. Okay, the ones that actually make the replication or the polymerization happen. So the DNA polymerase 3, this is sliding clamp loader, and then you have DNA polymerase 3, and you have tau proteins, which are flexible ones that are attached to the DNA polymerases like this. These two are going to work one after another on lagging strand. This would keep on working on leading strand. Leading strand does not require replace. So one Okazaki fragment is synthesized by this DNA polymerase, and then it will dissociate, and the next one will be taken up by this DNA polymerase. So I hope you remember the YouTube video that we have seen in the last class, but we can always watch it again. So it is kind of, 1 second, if you wait, I'll try to find it. 1 second. I'm trying to look if that video is present. Okay, maybe I'll try to find it some other time. But what here is something like this. This is the leading strand being synthesized. Okay, and here is the lagging strand. It is a complex picture. It's difficult to understand right away unless you have watched the animation. Hope you are trying to do that, and please follow the instructions to watch, to follow some of the materials before you come to the class, so that, and also read the textbook, and you'll feel comfortable with the course. So the green one shown here is one of the RNA primer, and then this DNA polymerase is synthesizing the lagging, the Okazaki fragment here like this. Okay. And what happens to the other one is this tau protein here. It has loaded the sliding clamp along with this is the loading sliding clamp loader. It has loaded the sliding clamp along with the DNA polymerase, and that is going to synthesize the next Okazaki fragment. This protein, this DNA polymerase, once it has reached here, it will dissociate from the sliding clamp and will get a new sliding clamp from here, and then will be attached to a new Okazaki fragment or new strand, something like this. Okay. Yes. So we will study some real biological way of seeing is. The first model was produced how DNA polymerase, DNA replication happens in this cell. It's called the Replicon model, which means that we know that replication does not start anywhere randomly. It starts at only one particular point, and that point is the replicator. And usually now we refer to this as the origin of replication. And there is a protein that recognizes the replicator, and that protein is called as the initiator. So there is a certain place where replication starts, and there are a group of proteins that recognize that particular initiation or the origin of replication, and that is how replication will proceed to produce two identical DNA molecules. So different organisms have different structures in terms of replicators. Here is ORIC, that is origin of replication. For E. coli, it has three. When you say 13 here, it means that is 13 base pair sequence. This is the same sequence repeated here 3 times. And there is another one that is 9 base pairs, 9 bases, which have the same sequence. And the total length is about 245 base pairs. Yes. So SV40, that's a virus, and it has a much smaller one. In all viruses, you'll find that gene density or everything is small, everything is tightly packed because it has to be in a very small size of DNA. We will read that in one of the lessons later. Saccharomyces cerevisiae, it's a fungi or yeast. It has about 100 base pair origin of replication. So if you look at different organisms, you will find different structures of replicators. And each of those replicators will have their corresponding proteins that recognize them. Okay, the proteins, the initiating proteins of yeast cannot work on E. coli, or E. coli initiating proteins cannot work on fungi. So there should be replicators and their corresponding initiator proteins. These two in combination will be able to initiate replication. So we will have a model that is best described for E. coli. I think the next slide should have, but okay. So the first thing that happens is there is a protein called DNA-A that recognizes the 9-mer. I hope you remember what is 9-mer. It's shown here. This is a 9-mer, right? There are 4 9-mers. That means 9 bases of the same sequence. DNA-A, when it is bound to ATP, it is in active state, so it will recognize the 9-mer sequence, and then it will coat the protein, and it will result in denaturation in this region, that is the 13. And once it is denatured, there is a DNA helicase loader along with DNA helicase. Okay, those are names you have to remember. First one is DNA-A. Then you have DNA B and C. C is the loader, B is the helicase, which will bind to the denatured DNA like this. You need two of these, right? One for each strand, and they will travel in opposite directions. So one of it is traveling in this direction, and the direction of movement is 5' to 3'. And they will keep on moving until they keep denaturing more and more DNA. As they're doing that, there is this protein called primase, which is the one that synthesizes the RNA primer, and it interacts with the sliding clamp and makes the RNA primer. That is shown in the green in this case, here and here. Then we have the holoenzyme, DNA polymerase III holoenzyme, with all its tau proteins, DNA polymerases, sliding clamp loader, and so on. So we'll also have 2 holoenzymes. I think we'll follow from here. So here they both are arranged, and then they will keep producing until they reach to the replication fork. And now you have a replication fork that will continuously work. And I hope you remember, why do we see this twist? That is this is called as the trombone model. The idea is to have, as I said, we should be able to work for direction of replication and direction of polymerization, right? This is how the replication fork is moving. And for this strand, the top strand here, no issues. I'll take this place. No issues, because the polymerization here is happening 5' to 3'. Okay, let me zoom it in a little bit. Yes, what is this? Okay, I hope you'll be able to follow this now. This is 5' to 3'. Right, so no issues. The direction of replication fork is this way, and polymerization here is also in this direction, so no problems at all. Look at the bottom strand. In the bottom strand, we have some problem. That's because we have to, replication fork is this way, and the template strand is in the reverse, or the direction of polymerization is in this way, right? So what it does is it twists the DNA in such a way that it goes back in this, it forms a loop like this. When it forms a loop like this, and this is the primer that you're seeing here, and the orange one is synthesizing. Because it is looped, or you can also consider it in a simple term. You can also consider it like this, like we have discussed in the last class. Say, if I twist it this way, okay, this is the leading strand, template for the leading strand. And leading strand would be produced in the direction, no problems. But for this, you will have a problem. So if this was the DNA polymerase, if this was the primer, when you put it three-dimensionally, you can put it in, you can also twist this a little bit. And in that case, you can have polymerization happening in this direction. Right? That's how DNA polymerase, that's how replication keeps happening continuously. We will try to watch the video or some other videos in the later class. I want you to watch the shared videos that I shared it on Google Classroom about, let me check if it is around. And if it is there, we'll play it. I hope you'll be able to see that here. Just give a minute. It also comes with an audio. I want you to watch this so that you'll find it comfortable. I have a question. Are you able to hear any audio? Are you able to hear an audio in the animation?
Not right now.
No, sir.
Okay, not even now, right?
No, sir.
I'll just to speak then. So DNA helicases bind, and then they keep separating the DNA strands using ATP. Then single strand binding proteins, they will, this is the replication fork that is organized. And then we will have the single strand binding proteins that will coat these strands here. So the job of the single strand binding protein is to prevent re-annealing or renaturing of the DNA. And we have topoisomerases because there is positive supercoiling developing here, because there is increase in twist and increase in drive, which will be solved by topoisomerases class topoisomerase 2, which makes double strand break. And then there is a primase, which synthesizes a small RNA primer, and then that is taken up by the DNA polymerase, and then polymerization happens. that is being the one that is being synthesized is leading strand, because the direction of replication fork and movement and the direction of polymerization is in the same way. Whereas the other one is produced in discontinuously, so that is lagging strand. So these are all enzymes that work very fast. DNA polymerase can add up to 1,000 bases per second. Its processivity is very high. That can be taken up, but the rate of polymerization is very high. And processivity can be increased by something like a protein called a sliding clamp, which will bind around the DNA, and then it increases processivity. So the RNA primers that are synthesized are not, or they should be removed, and that is where RNase H comes into play, and it can, or it can also be removed by DNA polymerase 1. I hope you remember Okazaki fragments. They could be, although they show it very small here, they could be something like 200 bases and so on, bases. Yes. The gaps will be filled up by enzymes like DNA polymerase 1, and then the mix will then be sealed by DNA ligase. So we have two DNA polymerases. There is the clamp loader, and these are the tau protein, and this makes a whole holoenzyme. This is something how it happens actually. But this is the schematic, right? So I hope you can understand that the looping is happening towards inside. The loop is going, the DNA is getting looped towards this side like this. Either way, DNA polymerase does not have to. It's all about three-dimensional things. If you can think of. Okay, I want you to look at the one that I have actually sent you. This is the animation video. It is in your Google Classroom. You can go there, and I have sent, these are animation videos. This is where you are usually go to classwork, go to Watson animations videos, click on that. And then this is what will open up for you. So there are 3 videos. If you want to, please note it down. First one I would want you to watch is DNA topology here. Second, you should watch about DNA polymerization, and then go to replication, the video that we have seen just now. Why I'm specifically going and kind of spoon feeding in a way is I want you to watch something for the next video, next class. That gets a little bit troubled because it's complex, and when you are able to watch, or when you watch the videos, you'll become much more familiar. Please watch this video and come. It's about telomerase. We will know, you should be able to understand what is end replication problem, And then we should also be able to understand how telomerase functions. So please watch this video for the next class. Otherwise, for tomorrow, I think we have in the first period. Please read it so that we'll all feel comfortable while we are reading. With the same lesson, this is what's about topoisomerases. This is also pertaining to the last in the last few classes we have been discussing about topoisomerases. You watch these videos, the audio, the narration on these videos is also fantastic. So I hope it will be helpful for you to read the textbook. At the end, the what will happen